Hi, how are you today? Today's lesson is about the antigen recognition by B lymphocytes. This is gonna be activity 6, I guess, page 125. The membrane receptors which are present or displayed at the surface of the B lymphocytes are said to be or called antibodies. If you take a look into this figure, you will notice that we have different types of antibodies. Each type can bind to a specific antigenic determinant, that is to mean a specific structure present on the antigen. This antigenic determinant is known as the epitope. So this antibody can bind to this epitope in a puzzle manner, in a key and lock manner. So they interlock with each other. So as you can see, this type of antibodies precisely can only bind to this epitope. It cannot bind neither to this nor to that. Similarly, for those two types of antibodies, each can bind to its specific epitope only. So this binding is in an interlocking manner. Which means that they bind with each other like in the case of a puzzle. An antibody has another name, which is the immunoglobulin. Usually it has a Y shape. It is a protein having a Y shape, and of course it's produced by plasma cells, which are active B lymphocytes. The antibodies neutralize the pathogens. Pathogens, when we say pathogens, they are actually non-self cells. For example, the bacteria, the viruses, the protozoa are all said to be pathogens. So when they enter to the body, the antibodies can bind to them and neutralize them. That is to mean it prevents them from targeting special cells and reaching into these cells. The antibody recognizes a unique molecule of the pathogen, which is known as the antigen. As we said before, it is actually the epitope of the antigen, to be more precise. Each tip of the Y of an antibody contains a paratope, which is representing a lock, and it is specific for one particular epitope which is present on the antigen, and it represents the key. This is why we say that they bind together in an interlocking manner and they look like a key and its lock. Here is the shape of the antibody. If you can notice, it has a Y shape. It is a Y-shaped structure, and by the tip of its Y, so it has two tips on this Y-shaped protein. By its tip, it can bind through, this is the region known as the variable region, which differs from one antibody to the other, and this is the site that it fixes. It uses this site to fix to the epitope of the antigen. So as you can notice here, according to the shape of this tip, it will bind to one only epitope of these antigens. So as you can see, because this is the only antigen that can bind or that can match with the shape of the paratope or the tip of the antibody, so it can only bind to this antigen and it cannot bind to the other types of antigens. The antibodies can occur in two physical forms. Usually the antibodies are present either in a soluble form, that is to mean they are secreted and released outside the plasma cell, and we can find them solubilized or located within the blood plasma, or we can find them still attached to the surface of the B cell. In this case, we call these antibodies B cell receptors when they are still attached to the B lymphocyte. The BCR or the B cell receptor is found only on the surface of the B cell. It facilitates the activation of these cells and their subsequent differentiation into either antibody factories, which are known as plasma cells. The plasma cells are the cells that can secrete the antibodies, or the memory B cells, which are the cells that remain for a very long period in the body to remember. The function, as the name tells, of these memory B cells is to remember the same pathogen or antigen if it ever enters the body again. This is gonna facilitate a faster response upon the second exposure, of course. Let's take a look into the antibody structure. First of all, the antibodies are glycoproteins, so they are made up of a mixture of carbohydrate and proteins, made up of two large heavy chains and two small light chains. The antibody usually has three segments, which are two variable regions present on the tip of the Y-shaped antibody, and a constant region, which is the region that fixes the antibody to the B lymphocyte. And between these variable and constant regions, there exists a hinge. Okay, so this makes the antibody made up of three segments. Although the general structure of all antibodies appear to be similar, however, the small region, which is known as the which is on the tip actually of the Y structure, this is known as the variable region, and it is what forms the differences between one antibody and the other. Okay, so this slight different tip structure are known as the structures, the variable regions that can bind to the epitopes of the antigen, and this is what makes the difference between one antibody and the other. As we said, this region on the tip of the Y protein or the Y antibody is said to be the variable region. Of course, this variable region differs from one type of antibody to another. As for the constant region, usually the constant region presents very slight variations. According to these variations in the constant regions, we can distinguish 5 different types of immunoglobulins. Remember that immunoglobulins is the other name for the antibodies. These immunoglobulins are IgM, IgD, G, A, and E. So there are 5 classes of the immunoglobulins, which are due to the difference in the constant regions. As we said before, the constant region is the region that fixes the antibody to the B lymphocyte. Let us take a look into the structure of the antibody. If you take a look into the antibody, you will see a Y-shaped protein. As we said before, it is made up of 3 segments. The first segment is the variable region. The second segment is the constant region, and these 2 segments, the variable and the constant, are connected by a hinge, as you can see. As we said before, there are four polypeptide chains constituting this antibody. These 2 chains, this is the first one, and this is the second, are said to be the heavy chains, while these small ones are said to be the light chains. So this is basically the structure of the antibody. Now let's take a look into the different classes of the immunoglobulins or the antibodies, which are different based on the differences, the slight differences in the constant region. So these are the variable regions of the protein of the antibody, which allow them to bind to different epitopes of different antigens. And usually the constant regions, basically they have very slight variations. Only these variations are going to allow us to differentiate between these different types of immunoglobulins. So of course all immunoglobulins that belong to the same class are going to have the same constant region, and the constant region is only different between the types of the immunoglobulins of one class if you compare it to the other class. Okay, so this is the IgG, this is the IgE, immunoglobulin type D. Immunoglobulins type M, as you can see, they look like several antibodies connected to each other. And this is the immunoglobulin of type A, which represent 2 immunoglobulins connected with each other. Let us examine some of the differences between these different types of immunoglobulins. As we noticed before, the immunoglobulin of type A, which looks like two antibodies connected with each other, usually these types are present within the mucus, within the saliva, the tears, and also present in the breast milk. So they are very important antibodies that move from the mother to her breastfed baby. And the function of such immunoglobulins of type A is to protect against pathogens. Immunoglobulins of type D are actually part of the B cell receptors. They activate basophils and the mast cells, and they are involved somehow in the allergic reactions. Immunoglobulins of type E are important in the protection against parasitic worms, like the worms that we can find in the intestines and or the liver and other organs. And they are very important immunoglobulins needed for the allergic reactions. Immunoglobulins of type G are secreted also by the plasma cells into the blood, and they have a very important property in crossing the placenta to the fetus. So in this case, when the mother produces antibodies, if she's pregnant, the antibodies of type G have the ability to move from the mother's blood to the fetal blood across the placenta. Okay, this is a very important property you shall know about the immunoglobulins of type G. As for the immunoglobulins of type M, they may be attached to the surface of the B cell, or they can be secreted into the blood, and they are responsible for early stages of immunity. We still have one objective to cover in this activity, which is the cross reaction. You're supposed to know that an antigen might possess more than one epitope, more than one antigenic determinant. So it can possess several epitopes. Each antibody is specific for a specific epitope of an antigen. That is to mean if the antigen possesses 3 epitopes, 3 types of antibodies can fix to this same antigen. Each can bind to its specific epitope. Also, when one antibody binds to 2 different antigens because they share the same epitope, here is where we talk about the cross reaction. So the cross reaction is the reaction performed between one antibody by fixing itself to 2 antigens because they share the same epitope. So if you take a look into the figure, here we have an antibody having a triangular shape, as you can see. This is an antigen having an epitope representing a triangle. So this antibody can bind to this epitope. And this is another antigen that possesses the same epitope. So because the same antibody is fixed to 2 different antigens, but because they have the same epitopes, this reaction is said to be cross reaction. Thank you so much.